Hi everyone, welcome back to class. Trust you all have been doing great. What we want to discuss today is the regulation of the Krebs cycle. The regulation of the Krebs cycle. And remember, in our last class, we discussed extensively on the Krebs cycle. We discussed about the pathway. We discussed about the intermediate, the direct and indirect product that we can get from the pathway, and the tricks and tips that we can use to know everything about that particular pathway. For those of us that have not checked out that class, I will leave the link to that class in the description so you can check it out. Now, moving back to what we want to discuss today is the regulation of the Krebs cycle, also known as the TCA. So, regulation of TCA cycle is what we want to discuss today. Now, before we go into that in detail, let's try and look at this metabolic pathway on its own and discuss the importance of this particular pathway. Number one importance of this particular pathway is that it is important in cellular respiration. It helps in the extraction of energy from nutrients. So it plays a very central role in cellular respiration. Another importance of this particular pathway is that it helps to generate energy. Remember, in our previous class, we talked about the generation of electron carriers that enters into the electron transport chain. So, this particular metabolic pathway helps to generate energy. Another importance of this pathway is that it helps in redox balance. That is, it helps in the oxidation and reduction balance in the system. Another importance of this particular pathway is that it helps in biosynthesis. In other words, we can say that it's important in producing precursors that you can use to synthesize other biomolecules such as amino acid and different biomolecules. Using the knowledge from our previous class about the tricks in knowing our TCA, let's do that to draw out our citric acid cycle now. So we said, officer, can I keep someone special for money? Let's now use this to write out our TCA cycle on its own. So, officer, that is oxaloacetate, can giving us citrates, I giving us isocitrates, keep alpha ketoglutarate, someone succinate CoA, special succinate, for fumarate, morning malate, then going to oxaloacetate. Do you see that it's not something so difficult? So we have TCA now. Remember, we said that the acetyl CoA that will start at as the starting point of this particular pathway could be gotten from different metabolic pathway. So let's say you have your acetyl CoA, and it is your acetyl CoA that will act with this oxaloacetate to start the cycle. Now, this acetyl CoA, we said we can get it from different sources. But for this class, we want to assume that we are getting our own acetyl CoA from pyruvate. So pyruvate is broken down to give us acetyl CoA, and this reaction from pyruvate to acetyl CoA is catalyzed by the enzyme pyruvate dehydrogenase complex. Now, let's now look at ways at which we can regulate this pathway. So our TCA can be regulated at 4 different points. We have this point where pyruvate is converted to acetyl CoA by the enzyme pyruvate dehydrogenase complex. That's one. It can be regulated here where this oxaloacetate with acetyl-CoA is converted to citrate, and by the help of the enzyme citrate synthase. That's number two. It can be regulated here where isocitrate is converted to alpha ketoglutarate, and by the enzyme isocitrate dehydrogenase. That's number 3 point. And the last point is at this point where alpha ketoglutarate is converted to succinyl CoA by the enzyme alpha ketoglutarate dehydrogenase. Let's now take them one after the other. But before we move on, whenever you hear regulation of any metabolic pathway, it's just talking about the process in which you can turn on the pathway or turn off the pathway. So when we say turn off, that means we are inhibiting the pathway. When we say turn on, on, turn on, that means we are activating the pathway. Do you understand? So anywhere you hear regulation of metabolic pathway, it's just talking about the inhibition of the pathway or the activation of the pathway. And in simple English that we do here is that inhibition means the turn off, you turn it off. The activation is you turn it on. So now, for the inhibition of this particular reaction, which includes the conversion of pyruvate to acetyl-CoA, where there is accumulation of ATP, is telling us that, remember that this pathway, everything about this pathway is just meant to give us energy. So when we now have accumulation of ATP, which is the energy, to prevent wastage, accumulation of ATP will inhibit the conversion of it. So when there is inhibition here, there will not be pyruvate will not be converted to acetyl-CoA. So indirectly, no acetyl-CoA to react with this, and this pathway is shut down. So now let's put inhibitor. We have ATP. Another thing that can inhibit this particular reaction is this particular product, which is acetyl-CoA, and that's an example of feedback inhibition, meaning the product of a particular reaction is the one inhibits that particular reaction. So we have a feedback inhibition. When there is accumulation of this product, it will inhibit this reaction from occurring. So there will be inhibition of this enzyme and it will not be able to catalyze this reaction. So another point is acetyl-CoA. Another thing that can serve as an inhibitor for this process is NADH, which is our electron carrier. When we have enough electron carrier, remember the electron carrier will enter into the electron transport chain to give us energy. So we have more than enough. There is no point for this process to occur again. So we want to shut it down. We want to turn it off. So NADH, which is our electron carrier, can also serve as an inhibitor for this pathway. So these are the three inhibitors for this particular point. Number two, what are the possible inhibitors of this particular reaction? Number one inhibitor of this reaction is ATP. Another inhibitor of this reaction is feedback inhibition by this product, which is citrate, meaning that accumulation of this product can serve as an inhibitor for this reaction. So we have citrates. Another inhibitor of this particular reaction is succinyl-CoA. And lastly, the other inhibitor for this reaction is electron carrier NADH. So these are the inhibitor for this particular reaction at point 2. Number 3 now, in conversion of isocitrate to alpha ketoglutarate, it can be turned off when there is ATP. So meaning that we have enough energy, stop working. And also when we have electron carriers, NADH, they can serve as a regulatory molecule to this. So we have inhibitors there. Let's move on to the fourth point. What can serve as inhibitor at this particular point? One, presence of ATP. Accumulation of ATP can serve as an inhibitor of this reaction. So we have ATP. Two, electron carriers, NADH, can serve as inhibitor. Three, succinyl-CoA, which is the product of this particular reaction also, can inhibit this particular reaction. So we have succinyl-CoA. The work of all this inhibition is that they help to stop this reaction from happening. Let's now look at things that can activate this circle, that can turn it on. One, ADP. ADP can turn on this particular reaction. Another thing that can turn it on is NAD plus. NAD plus can serve as a turn on to this reaction. Then another thing that can also turn it on or activate this particular reaction is calcium, and that is when there is contraction of the muscle. So it's also signaling to the pathway that working very well. I need you to keep on this pathway. I need this pathway to keep going. So calcium can serve as an activator of this reaction also. Pyruvate can also serve as an activator of this particular reaction. So for the first one, which is conversion of pyruvate to acetyl-CoA, our possible activators are one, pyruvate, ADP, NAD plus, then calcium. Now let's move to number two. Activator of this reaction here is ADP. Meaning that when there is presence of this ADP, it is telling the pathway that I am actually low on energy. I am low on energy. I need you to start producing more energy for me. Activator of this particular reaction. Let's move to number three, which is the reaction that involves the conversion of isocitrate to alpha ketoglutarate by isocitrate dehydrogenase. Number one activator of this reaction is ADP. ADP. Another activator of this reaction is NAD plus. NAD plus. The other activator of this reaction is calcium. So three of them like this are the activator of this particular pathway. We have the ADP, NAD plus, then the calcium. Now let's move to the last one, which is the conversion of alpha ketoglutarate to succinyl-CoA. Number one activator of this reaction is ADP. And another activator of this reaction is calcium. So these are the possible activation of this particular reaction. Let's do a summary of everything we've been discussing in today's class. Regulation of the Krebs cycle, also known as the citric acid cycle, also known as the carboxylic acid cycle. So then the first place is at the conversion of pyruvate to acetyl-CoA, and the enzyme that catalyzes that reaction is pyruvate dehydrogenase complex. Then the number 2 point is that the conversion of oxaloacetate to citrate, and the enzyme that catalyzes that reaction is citrate synthase. Then the third one is at the point of conversion of isocitrate to alpha ketoglutarate, and the enzyme that catalyzes that reaction is isocitrate dehydrogenase. While the last one is the conversion of alpha ketoglutarate to succinyl-CoA, and the enzyme that catalyzes that reaction is alpha ketoglutarate dehydrogenase. So these It's just the summary of everything we've been discussing so far in today's class. By this, we've come to the end of today's class. See you in the next class. Bye bye.